come together to try and see how we can solve one of the problems that affects cassava, and that's a disease, and seeing how we can come up with, use the modern science in solving that problem. The two diseases that we are having, one of them, the cassava mosaic disease, it's spread in different parts of the world. You'll find it in where cassava came from in South America. You'll find it in Africa, in different parts of Africa. You'll find it in Asia, and that is cassava mosaic disease.
Yes.
But when it came now to East Africa, it found another problem. We still have not been able to establish exactly where the disease came from. This is cassava brown streak disease.
Okay.
So because it is not anywhere else in the world, it's only in East Africa. So because it's only in East Africa, for many years the disease was restricted to the coastal region. So, but around 2000s, we started realizing that the disease has come up to the highlands. But the cassava brown streak disease is a problem that has no solution as yet. And after looking at all the places that we could think for resistance, we didn't find any. So, but the good thing was that we had the modern science, what you call biotechnology,
yes.
And one of it, what you call genetic engineering, could help us to try and see, just the way we do the immunization, you get a virus and you inject into an animal, it produces antibodies, and those antibodies can be used to help in terms of creating immunity to other organisms. If you look at the genetic makeup of an organism,
yes.
we have what we call the DNA.
Yes.
The DNA contains the genetic information, the one that is translated to what we can see. So there is genetic information, and there is what you call the genome that has the information, and that gives what you call the genotype. That is the genetic information. When it is translated to what you can see, we call it the phenotype, or what you can be able to see. So that information that from the DNA is the one that defines how an organism looks like. So if you can only add, if that plant is infected with a virus,
yes.
the information from the organism is from DNA goes from DNA to RNA to proteins. So in that process, if there is a step, RNA, we say is single-stranded because
yes.
it's only picking the information from the DNA that is double-stranded,
yes.
and that information, the specific information that is needed to make the protein that is needed for phenotype, for what you can be able to see. So in the process, if you have RNA that is double-stranded, because RNA is supposed to be single-stranded, it's only getting the information that is needed.
Yes.
You don't have it to be in duplicates, or you only need it, the information is one. So it is brought in like a single page. So if you make them to be double, nature does not want that. When the plant realizes that, and any other organism, it starts cutting double-stranded RNA into pieces. So if you create double-stranded RNA structure, the organism cuts it into pieces, and those pieces are used as signals to the rest of the plant and to the rest of the organism to tell them that there is an organism that is not needed. There is a sequence that is not needed in the body. And that's what creates those signals. Then that mechanism, those signals, we call them dicers, and it's still science that we may not go to for now. That signal is transferred now to the rest of the organism and says that if you find this sequence, this combination, you know it is not needed, and it's cut into pieces. And what we are saying is, because RNA has been stopped from moving to the next level of producing what you can see, which is a part of the protein that is now transformed into what you can be able to see through other mechanisms.
Yes.
If you stop that process of going to the protein, that means that you have silenced it. And that's why we are calling it post-transcriptional gene silencing. So the information from the gene has been taken from the DNA
yes.
through transcription.
Yes.
It comes to an RNA, and that RNA is one that is supposed to be translated to what you can see as a protein. So we are calling it post-transcriptional because transcription has already occurred.
Yeah, okay.
And it's going towards, now we have the RNA.
Yes.
RNA, now we create a double-stranded RNA that makes it now to be cut into pieces, and it creates a signal. It's a normal process that occurs in every organism. If it's affected by, if it's double-stranded RNA is created, the silencing starts. You remember that the virus and the plant in this case, they are still, or both of them are trying to coexist.
Yes.
So it reaches a balance where the virus is not killing the plant, but it is still surviving within the plant. So in some cases, the plant may become resistant because it has been able to keep silencing that virus that comes. The virus, by the way, most of the viruses are RNA.
Yeah.
And majority of the viruses are single-stranded RNA. So since that one now if we create a double-stranded RNA using the virus itself, we say that you have the complementary strand of a similar strand that has the complementary of the nucleotides that are in the RNA. That now when you create that complementarity, that cuts that into pieces. So now what we did is to enhance that process by creating that double-stranded structure. And that signals the plant that if a virus comes with that similar sequences, then it attaches itself, it looks for that, it attaches itself and starts the process of silencing.